A very good afternoon and welcome to all. Myself, Dr. Amsaveni, working as Assistant Professor in the Department of Biotechnology, Kongunadu Arts and Science College, Coimbatore. On behalf of PG and Research Department of Biotechnology, I feel happy to join with you all to host this third day International Alumni Webinar Series on Insights into Translational Research and its Applications. With all your cooperation and constant involvement, as well as the moral support, guidance, and motivation of our inspirational women, Madam Dr. C.A. Vasugi, Secretary and Director of our esteemed institution, we have successfully completed 2 days of this webinar series with our prestigious alumni as resource persons. With this note, may I now call upon Dr. V. Bhuneshwari Ma'am, Associate Professor and Head, UG Department of Biotechnology, to offer the welcome address and introduce today's resource person, Dr. M. A. Shibu sir. Ma'am, please.
A very good afternoon and hearty welcome to all on behalf of Department of Biotechnology for the third session of International Alumni Webinar Series on Insights into Translational Research and its Application, which is organized between 3/6/2020 to 8/6/2020. First of all, I dedicate my welcome in absentia to our beloved President Dr. M. Archami sir and to our beloved Secretary and Director Dr. C. A. Vasugi ma'am for their immense support and encouragement rendered in the growth of this department. I warmly welcome our dedicated and sincere principal, Dr. M. Lakshmanaswamy sir, who is our HOD previously when the resource person and me joined in this department of Kongunadu Arts and Science College. I gladly welcome sir. I also welcome our distinguished and enthusiastic Chief Executive Officer Unaided Courses, Dr. V. Chinnusamy sir. I welcome you sir. I express my delighted welcome to our respected and motivating Dean R&D, Dr. S. Paulsamy sir, and supporting Dean Academics cum HOD, Biotechnology PG, Dr. S. R. Madan Shankar sir. I welcome you sir for their constant support and immense help rendered to for all our endeavors. Now I cordially welcome our guest of honor, Dr. M. A. Shibu. I welcome you sir, who readily accepted our invitation in spite of his busy schedule for this program. I'm really proud that my classmate is working in a top 1 hospital in Taiwan with 13th rank in world level. I kindly welcome all the participants for their interest for this program. I warmly welcome the organizing secretaries, Dr. R. Amsaveni and Dr. M. Gokul Ramnath, Assistant Professors in Biotechnology. I welcome you, ma'am. I welcome you, sir. Session coordinators, Dr. P. Sendil Kumar, Dr. G. Anbarasi, Dr. R. Ranjit Kumar, Dr. B. Vishnupriya, Assistant Professors in Biotechnology, other staff members, research scholars, student participants from different institutions in abroad as well as in India. Once again, I welcome you all. Now it's my pleasure to say about the resource person, Dr. M. A. Shibu, who has studied both BSc and MSc in Kongunadu Arts and Science College during 1999 to 2004. He has completed MPhil in Biotechnology at Bharathidasan University and PhD in Biotechnology at National Dong Hua University, Taiwan in 2012. He has completed his postdoctoral research in the Graduate Institute of Basic Medical Sciences, China Medical University. His research specialization includes molecular and regenerative medicine with particular focus on cellular and subcellular events. He is currently acting as assistant researcher, principal investigator in cardiovascular and mitochondrial related disease center, Buddhist Su Chai Medical Foundation, Taiwan. He is acting as a PA for 3 projects and completed 3 projects. To his credits, he has received Taiwan Ministry of Science and Technology Postdoctoral Award 2017, CMU Asia Postdoctoral Fellowship 2013 to 17, Taiwan Ministry of Foreign Affairs Research Fellowship Award 2008 to 2011, and Tamil Nadu State Council for Science and Technology Research Award 2003. He is acting as a reviewer and editorial member for several journals. He has published 31 research articles with cumulative SCI impact factor above 150, with citations more than 600, H-index 16, I-10 Index 25. He has acted as an invited speaker for 3 international conferences and presented 7 papers in conferences. With this brief introduction, now I invite Dr. M.A. Shibu, the Principal Investigator in Cardiovascular and Mitochondria-Related Disease Research Center, Buddhist to Child Medical Foundation, Taiwan, to present his views on the emerging topic, stem cell therapy for aging-associated disorder. Sir, please.
Thank you, ma'am, for welcoming the participants and introducing our resource person. And before starting the session, our Dean Academic Sir has joined. Sir, do you want to share some words? Dr. S.R. Madan Shankar, sir.
Yeah, good afternoon. I'm here.
Good afternoon, sir.
Good afternoon. Good afternoon to all of you. Dr. Shibu, Dr. Bhuneshwari, Dr. Amsaveni, and all the faculty from the Department of Biotech. It's nice to see our alumni blossoming well in different places. I congratulate Dr. Shibu personally for having achieved great laurels in this particular field. That is stem cell research, which is going to be one of the track changer for most of the diseases which we are encountering in the current day. I would always say that this college, that is Kongunadu Arts and Science College, and the Department of Biotech has given us, including myself, I am also an alumni, including myself, has given lots of basic stuff so that we are able to do cutting edge research in our own chosen domains. I'm happy that you have accepted to be a resource person in our webinar series, and on behalf of the management and principal and the department, I once again welcome you for this session. Also, I welcome all the participants and congratulate everybody for having joined us and supported us in this endeavor. Thank you.
Thank you, sir.
Thank
Now,
you.
now I request Dr. M.A. Shibu, sir, to take over the session.
Good afternoon, all. Hello.
Good afternoon, sir. It's audible, sir. It's audible, sir.
Good afternoon, all, and thank you, Dr. Madan Shankar, for thank you, Dr. Madan Shankar, for the introduction. I also thank the Secretary, Director, Dr. Vasuki, and the Principal, Dr. Lakshmana Swami, and the conveners, Dr. Madan Shankar and Dr. Bhuneshwari, for this opportunity. I take this up. Am I?
Sir, it's not audible.
Sir, it's not audible, sir.
Hello, Bhuneshwari. Hello.
Sir?
ஒண்ணுமே இல்லையே. Hello.
Okay, sir. 1 minute, sir. There is a technical problem. I will present it, sir. I will present the.
Okay. You present the screen.
Hello, Dr. Shibu, sir.
Sir? Shibu, sir? Sir, Dr. Shibu, sir? Sir, please unmute your audio. Dr. Shibu, sir?
Hello.
Hello, sir.
Madam.
Sir,
இன்னும்
please.
ஆடிபிளே இல்ல. அதாவது ஸ்பீக்கர் செக் பண்ணா சரியாகும்.
Okay, sir.
அவங்கள ask him to check the speaker
Okay.
settingsல.
Dr. Shibu, sir? If you have problem in presenting me, I am presenting the slide. Please give audio alone. Please unmute your microphone. Sir, Dr. Shibu, sir, I am proceeding. Please unmute your audio and talk.
Am I audible now? Yes.
Yes, sir.
I'm sorry. Sorry for okay. So sorry for the thank you for the patience and the here I'm here to present like little bit insight on stem cell transplantation for aging disease. I work after finishing my master's and bachelor's. I came to Taiwan for my PhD, and after that, and after my PhD, I just continued as a postdoc here, and then I work in Buddhist Sichi Hospital, which is a part of Buddhist Sichi Foundation, which is established all over the world, and is well known for stem cell transplantation therapy. This hospital is one among the 4 hospitals that is approved for stem cell transplantation in Taiwan. And here we do stem cell transplantation for we have now approval for clinical trials for neurodegenerative disease, and we have approval for performing treatment with the hematopoietic stem cells. Yes. So this is the status of the hospital, and in our team we do some preclinical tests on improving the stem cell potential, particularly for aging and cardiac diseases. Okay. So 1947, you may know, 1947 is an important year for Indians. Like India, every Indian will remember it because that's a year for independence. But similarly, 1945 is another important day, a hallmark in the history. That's because the World War II ended in 1940 or 1947. But that was a devastating year for people from Hiroshima. That could be because that's because of the nuclear bomb blast. Although there were survivors, the people were affected and mostly with blood diseases. And scientists were scrambling to find a treatment for this. And one physician, hematologist, like Dr. Lawrence, he tried to find a way to using hematopoietic stem cells transplanted into patients that are immunocompromised to boost their immunity. And this was the first step on stem cell transplantation. And curious scientists, they just catched up and tried to formulate strategies and improved upon this. And in 1968, after the discovery of bone marrow stem cells, successfully 2 children received bone marrow transplantations in 1968, and that appeared successful. But still, after that, there were a lot of failures in this therapeutic strategy, and that could be because of the graft rejection. When we transplant stem cells into a host that will be immunologically rejected. And this was an issue that carried about almost a decade. And then scientists just got up, they found strategies, they found new types of stem cells to come over these issues. And so this is the way that could many scientists and many physicians, they foresee that this could be a very very future of the therapeutics, personalized therapeutics that could improve the health, that could improve health in the society. Healthy society. When we talk about healthy society, we say that the increase in lifespan will indicate as an indicator of healthy society. In 1947, if you see the average lifespan of the globe. In the globe, the average lifespan globally was below 40. But now, in the year 2019, it has increased up to 60, more than 60. Only if selective, just 4 or 5 countries have life expectancy less than 60. So in a interesting study in United Kingdom, particularly on based on the data from England and Wales, they show that about 40% of the population will be over 100 years in the year 2013. This is so interesting but so concerning as well, because when the aging population grows, about 40% of the people, if they are over 100 years old, that causes a concern in gerontologists, because aging is sometimes very scaring because not because people get aged, like people don't get scared because of aging, but because of the sufferings associated with the aging. Aging, they cause they cause physiological dysfunction. They reduce the physical function, cause a lot of diseases, and also they cause cognitive and physiological disorders. And there's a term for the scare of aging called gerosophobia. It's so now the understand of, like, about a decade ago, the healthy society was literally meant that the life expectancy will be high. But now we are term health span. Now, more than the lifespan, the health span of the individual is more important. We live 100 years, but how long we live healthy? That's a concern now. Okay, so with aging, there are a lot of diseases associated, like Alzheimer's, like disorders in the vision, heart, and also the like metabolic disorders, like problems in the bone and like vascular systems. So this could be because that humans, by evolution, humans have lost their potential to regenerate, or regenerative potential. This is because like the primitive organisms that stay still, they have their regenerative potential restored. But humans doesn't have that much. Like in the case of axol, the even if their limbs are lost, cut off, they can develop with their muscles, with their bones. Yeah, that's because they can de-differentiate. Any cell in them can de-differentiate and form a like yeah, and can regenerate itself. So what is the problem with humans? At younger stage, we see the abundance of stem cells, but with and healthy stem cell pool. But with growing age, we see that the stem cell pool reduces. The amount of stem cell that is available for regeneration is reduced. So when there is a damage in an organ, the stem cells usually just replace the damage or just by differentiating. But this doesn't happen when we get aged, because not only the stem cell pool reduces, but also the stem cells lose their dysfunction, and also there'll be some mutations in the stem cells. The mutations causes like defects like cancer, and also dysfunction. Dysfunction causes degenerative disorder. When there is damage or degeneration of tissues, the stem cells may not be potential enough to replace them and to repair them. So, what is the current scenario of stem cell therapy for aging-associated disorder? Is it like just taking some cells from the donor and then injecting those young cells into the patient, elder or aged patients? It looks so simple, and so that was a case of allogenic transplantation, taking stem cells from a donor and then injecting it to an elder or a patient to improve their potentials. But in another case, autologous transplantation. In autologous transplantation, yes, from stem cell from the patient will be taken and will be activated or potentiated so that they get better regenerative potential, and then again will be transplanted back to the donor, the patient, so that they for a therapeutic purpose. So who are the targeted patients? So who are the targeted patients for aging treatment? That's very obvious that elderly patients like above 60, and also people who are inherited, like they have genetic problems associated with aging. So these are the target patients that are very obvious patients. But usually these patients, we cannot identify them at an early stage. For example, those who live with their grandparents or like elder people will know that they live with us with like we will find that they have aged only when we start finding that they forget words, they forget stuffs like their regular work, they decline in their function. Only based on that we find that they aged. But yeah, like there are other people, like for example, I still remember when I was a student in Kungunadu, I used to climb, like my class was in the fourth floor. We used to climb up and down multiple times to come to lab, to go to the class like that. But I never gasped for breath. But now I don't think I can do. I'm around 40. This could be because I'm aging. Yeah. At 40, the process of aging starts after 20. So many people are susceptible for aging. For example, those who have busy schedule, those who don't have time to relax, they are susceptible for aging associated disorders. And also people who work very hard, who play very hard, meaning they have a lot of recreational activity, very like recreational activity, and they may have addicted to like drugs or smoking, alcohol, and they also have they are very much susceptible for aging associated disorder. In addition, athletes tend to cross the barriers of human potential. They do so. This also affects the person and cause aging associated disorders. So aging like treatment with stem cell is not just for elderly patients, but also for susceptible younger patients as well. So what are the stem cells that are now considered? Like now, basically the embryonic stem cell and adult stem cells like mesenchymal and tissue specific stem cells like neuron specific stem cells, or even hematopoietic stem cells, all these. And mostly now, most of the researchers are trying to find some treatment modalities with mesenchymal stem cells derived from bone marrow, or umbilical cord, or adipose, or placenta. And okay, so when we use embryonic stem cells, are very efficient, and they can be acquired in abundance. But still, there are a lot of problems, like the differentiation. Once they have very good differentiation ability, actually, but when transplanted, the differentiation efficiency get lost, and they undergo teratoma formation, and also they have inappropriate migration. Teratoma formation meaning like they form tumors when engrafted, when transplanted, they cause tumors. Also, inappropriate migration meaning when you inject stem cell for cardiac treatment, they may go to liver. And induced pluripotent stem cell. So one of the problem associated with the stem cell transplantation is like when once transplanted, the host may reject it. So to like iPSC would give like using induced pluripotent stem cell would give a solution for this, because iPSC can be derived from the patient's cells. Like the patient's adult cells can be de-differentiated into a stem cell, or a dysfunction stem cell can be made into function, like more efficient or potential stem cell for regeneration. The first iPSC was generated using a mouse fibroblast, which is which is proliferative but still adult cell. In this cell, they use some transcription factors from stem cells like OCT4, SOX, KLF4, c-MYC. They are transcription factors which helps to maintain the stemness of a stem cell. Stemness of stem cell is stemness of stem cell is determined by so many factors. The transcription status of the cell that controls the differentiation, de-differentiation of the cells. So, but these transcription factors, for example, the c-MYC, when c-MYC is overexpressed in a stem cell, they forms cancer. They divide unlimitedly. And also, when these transcription factors are overexpressed via retroviral mediated vectors, these retroviral mediated vectors, they also can induce the proliferation of the cells. They bind to the DNA and the integrative plasmid, and it can also induce cancer in the cells. So this is and then so now mostly they concentrate on somatic. Apart from induced pluripotent stem cell, they can also concentrate on somatic stem cells, which hematopoietic stem cell and mesenchymal stem cells are of the good options. And hematopoietic stem cells are derived from bone marrow, peripheral blood, umbilical cord, and mesenchymal stem cells from dental pulp, bone marrow, adipose tissue, umbilical cord. I'll explain that in detail. Like hematopoietic stem cells are generally used for transplantation for immune dysfunction, to treat immune dysfunctions, and also they are known to enhance neurogenesis and angiogenesis. They produce some factors that can enhance the neurogenesis and angiogenesis in cells. Neurogenesis means genesis of neuron cells. Angiogenesis means production of new blood vessels. And but also these hematopoietic stem cells are associated with malignancy, and because of the immunological feature, they destroy they interrupt the immune system and cause autoimmune disorders, and thereby they also affect the endocrine system. They produce a lot of unwanted endocrine signaling. They disrupt the endocrine signaling. Also, umbilical cord derived mesenchymal. This is more, it's very attractive now. For example, in our hospital, we use Wharton's jelly mesenchymal stem cell. It's also an umbilical cord stem cell, and they are very good because they have very good differentiation ability, and also they can they have paracrine activity. They produce synthesis lot of growth factors, neurotrophic factors So they are very attractive for the for stem cell therapy research. And bone marrow derived cells, they can be either a mesenchymal stem cell or hematopoietic stem cell, but bone marrow derived mesenchymal stem cells have shown a lot of success so far. But the problem is for example, in the heart disease and heart disease, they show a very good treatment with bone marrow derived stem cell give good improvement for the cardiac contraction and relaxation, cardiac function. But still, there is an issue with bone marrow derived mesenchymal stem cell because they interrupt the cardiac pulse. They also have calcium signaling. Usually cardiac cells, they contract and relax based on a calcium signaling mechanism. But bone marrow derived mesenchymal stem cells can affect that. So this may treatment with bone marrow derived mesenchymal stem cell can affect the cardiac function. And another one is adipose derived mesenchymal stem cell, which is recommended by many researchers now, and they also have very good differentiation as well as paradigm expression. Then, then is uh, but the difference, like as I told, bone marrow derived mesenchymal stem cell and adipose derived mesenchymal stem cells are very attractive, but there is a difference. Bone marrow mesenchymal stem cells in the bone marrow, they reduce with age. When people get aged, the population of the mesenchymal stem cell in the bone marrow reduces. Like from 10,000, it goes to 1 in 2 million. But adipose derived stem cell are not so. For example, the total population of adipose derived stem cell will be like will be reduced upon age, but still it's not that much. Just there will be 80% of retention of stem of mesenchymal stem cells in adipose tissue. Also, like for example, in just to demo, just to say about the stem, like to explain about the efficiency of the stem cell therapy, I take neurodegenerative disease as an example. Like as you know, aging, neurodegenerative disease are very much associated with aging, and Alzheimer's disease is one of them. Upon aging, there's a loss of neocortical and archicortical volume. Neocortical volume is related with sense like smell, taste, this and vision, and archicortical volume is associated with archicortical volume is associated with memory. So when these volume reduces, the memory like memory loss will be obvious. Also, there'll be a decrease in brain weight. With aging, there'll be the enlargement of the ventricular system. Ventricular system contains the enlargement of ventricle system it's an obvious one, and it contains a CNF fluid in it. Also, the neurotransmitters receptors also they decrease. This means that the cell-to-cell communication or the signaling between the neurons will be also declined. And altogether, there is a reduction in white matter and gray matter. For example, here you can see the loss of gray matter. These three are the panels show the your brain mapping by fMRI scanning. They show the yellow color indicates gray matter. The amount of gray matter in younger patients, individuals, they're high when compared to the older group. This shows the aging affects the gray matter level in the aging. But in Alzheimer's, even if it is a younger patients or the older patients, we see a reduction in gray matter. Okay, so in Alzheimer's disease, the cause of Alzheimer's disease is because of two proteins, one is tau and amyloid beta. These two proteins, they misfold, and because of misfolding, they form aggregates. These aggregates, because of aggregation, the cells undergoes massive stress, and because of that, the cell death occurs, and cells dysfunction and cell death occurs. Loss of neurons will be the obvious result. And because of the tau protein, they form aggregates. And for example, this is a smooth. This figure shows a smooth appearance of the neuron cell, but with misfolded aggregates of tau protein, they form tangles, neurofibrillary tangles in the cell. And also the amyloid protein, they form they misfold and form plaques, and these aggregates they disrupt the signaling between the cells and also cause dysfunction. So when you treat mesenchymal stem cells, then we transplant mesenchymal stem cell cultured in laboratory conditions and into the directly into brain. The expectation is that the plaques and the tangles will be reduced, or these tangled or cells that produce misfolded or aggregated amyloid beta will be removed or replaced by the new cells. So there are so many clinical trials which are going ahead in this aspect. Like there are 3,939 clinical trials on aging associated diseases, and about 35 clinical studies that are conducted now just for against the aging induced susceptibility. Yeah. So this seems to be easy. Like it seems to be very cool, and we feel that obvious results will be achieved, like they significantly like this. But there are major clinical hurdles. Major clinical hurdles, like when, as I told, like when we inject a stem cell in 2 days. In the 2 days, we have experienced loss of 90% of the cells that we have injected. Only 2 to 5% remain after a week, and no cells will be there after 4 weeks. This is a very big challenge. And also, we don't know, after injecting, we don't know what happens to the cell actually, and how it disappears, we don't know. And then also poor survival. Like when we usually use stem cell to treat diseases, the disease conditions will have greater stress, and when we inject these stem cells in this very susceptible environment, the stem cells undergo stress and they don't survive well. And also major safety concerns, like when we inject stem cells, we have like teratoma formation, the tumor risks also. And moreover, we have economic and ethical hurdles as well. So to overcome these hurdles, we need to find how these stem cells function. So usually when we transplant a stem cell, they just elicit their effects in 2 ways. One is by paracrine effect. They secrete growth factors, cytokines or immunomodulators, and these growth factors can help the damaged tissues to just self-renew themselves, or they just move to the damaged tissue and differentiate into the tissue types. For example, if we have to cure cardiac damages, we inject stem cells, and these stem cells move to the damaged area, and then they differentiate and replace the damaged tissue. But what are just like I mentioned, the poor survival and poor homing, poor differentiation are all the hurdles for this. So once we, so in order to overcome this, we concentrate more. Like many people, they concentrate on homing. When we inject stem cell, the stem cell should find the place where the damaged tissue is there and should move towards the damaged tissue. And this is known as homing. The stem cell moving to finding the damaged tissue and return there. That's called homing. So and we also does. So when we inject these stem cell, can this homing or the potential of the stem cell affect because of aging stress? Yes. So far we were talking about the aging disorders, and we have a weapon with stem cell to treat that. But actually, the main issue is the stem cells. They undergo aging particularly when we use the stem cells to treat aging. So there are 2 aspects on this. One is the mitochondrial function. Usually the stem cells, they are in a resting stage or a quiescent stage. So their mitochondrial function is very low. They don't, like, their metabolism is not like, they have a very low metabolic level, and so the mitochondrial function is actually not so high. So they don't have the RO stress in the beginning, like in the quiescent stage or the resting stage. So, but when the stem cell is potentiated, when there is a damage, the stem cell should just move to the damaged area, and then it gets potentiated. For that, it gets potentiated, and then they are if they have to proliferate, they have to synthesize more protein all of a sudden. And because of this, the proteins synthesized by them in that cells will not be folded properly. These unfolded proteins, there'll be a lot of accumulation of this unfolded protein which are not properly folded, which doesn't, because of unfolded nature, they cannot function properly, but they form aggregates. When they form aggregates, they cause like a lot of stress in the cells. They induce ROS. They also, because of that, the cells dysfunction, and stem cells, they eventually die. So, so what are the means to overcome these two challenges? This is what we are doing in our lab to overcome these two challenges. So we have found, like generally, there are a few strategies that are used, like the preconditioning of stem cell before the therapy. We precondition the stem cell with hypoxia, meaning like we culture the cells in low oxygen condition to adaptation in a to get adapt to a stressful condition. And then we treat the cells with pharmacological agents or the agents that promote the longevity or survival of the cells, and also can modify the genes so that they promote the regenerative or the therapeutic potential of the stem cells. Particularly, we focus on hypertension. Hypertension is, as you know, it's associated with aging, and it is responsible for 7.6 million deaths per annum. It's about aging and hypertension. As I told, the aging and hypertension are interlinked. Most of the almost all aged upon aging, the blood pressure level increases. All aged individuals will experience hypertension. That's because their vessels, their blood vessels, they constrict upon aging. So usually, when in hypertension, the the heart, they remodel meaning like the heart overworks. The heart undergoes damages. As you know, heart is consists of cardiomyocytes, muscles that are mature. They are very much matured cells. They cannot differentiate or renew themselves. So when the cells die, they cannot be there are so many researchers that shows that there are cardiac stem cells, but later they have proved that it's like there are no cardiac stem cell. There are so many controversies on that, but that's different. But when a heart tissue that gets damaged, they will be repaired by fibroblasts. By filling with the collagen, the damaged area will be repaired. But upon aging, this repair mechanism is not regulated. So the cardiac, the accumulation of collagen in them will exceed a level so that fibrosis occurs. Fibrosis is even like when a heart undergoes fibrosis, the collagen accumulation will be high on a damaged area, so that the cardiac contraction or the relaxation function, cardiac function will be reduced. The heart will not have that elasticity to contract and relax. So this phenomenon is enhanced by age, like will facilitate by aging. For example, when the athletes can run faster. When they run fast, their blood pressure increases, but that doesn't affect them because even if there's some damages in the vessels, it will be healed by their regenerative system in them. But in aging, it's not possible. So aging people are more affected by hypertension. So this is obvious here when you see in a young patient, usually the wall upon hypertension, the cardiac wall thickens, meaning hypertrophy. So when usually hypertrophy is a first response of the heart when to stress. When there is a stress, the heart has to overfunction. So when the heart has to overfunction, they adapt themselves with the thicker wall to function more. But when the wall is too thick, they cannot. The contractile function of the heart will be lost. For example, in the case of aging, the cardiac wall is very thick. The hypertrophy level is so high that it goes to a pathological condition, and the cardiac function will be lost. Yeah. So what we do to overcome these challenges with the stem cell? So we try to in our lab we do preclinical studies to precondition the stem cells to increase the mitochondrial stress. And also we we try to enhance this homing of the stem cell by preconditioning, by finding some better preconditioning methods. Also, we try to find some methods to reduce the proteotoxicity and maintain the proteostasis in stem cells, and also try to protect the stem cells. I'll just show you some examples here. So how we see that hypoxia preconditioning of stem cell can improve the stem cell potential. So usually we see that in younger we have used a hypertension model, a rat model here. In this rat model, the stem cells collected from the adipose tissue show a reduced mitochondrial arrows in that. Reduced can be seen from reduced red color fluorescence. We have stained the cells with mitosox staining. This mitosox staining will show the mitochondrial ROS. So increase in mitochondrial ROS can induce the differentiation and death of stem cells. So in younger SHRs, you see that the mitochondrial ROS is less, but in older SHR is a spontaneously hypertensive rat. In that, the adipose-derived mesenchymal stem cells, they show increased mitochondrial ROS. So what we did, we tried to find hypoxic preconditioning to increase the stem cell potential. Stem cell potential, we say stem cell, how we determine stem cell potential is here particularly we say stem cell potential based on the stemness of the stem cell. The stem cell, like nano, is a stemness marker, transcription factor of stem cells, and P21 is a cell cycle regulator. This cell increase in P21 means arrest of cell cycle. So we found a different timing of hypoxia, and we found that 6 hours of hypoxia can very well maintain the P21 level below a threshold, and also can enhance the nano, which is the transcription factor important for stemness. So we thought 6 hours of hypoxia will be better than 24 hours of hypoxia. Usually, many other labs, they use 24 hours of hypoxia treatment. And we feel 6 hours of hypoxia will be good enough. So that was seen also here, like the 6 hours of hypoxia, they actually in fact reduce the mitochondrial ROS. But 24 hours of hypoxia, they increase the mitochondrial ROS. So 24 hours of hypoxia has a risk of increased differentiation. Increased differentiation relates, like when there is more differentiation, the immune system recognizes it and the cells will be lost upon transplantation. So we use the 6 hours of hypoxia preconditioned stem cells, and by tail vein injection, we just transplanted these stem cells into the rats. And we found that this upon transplantation, they gave a very good cardioprotective effect in spontaneously hypertensive rats. Okay, and then we also check the tail vein blood pressure. So upon, like usually in hypertensive animals, the blood pressure, both systolic and diastolic, are high. But upon stem cell treatment, there is a considerable reduction. Not so significant, but still there is a reduction. But the cardiac, this is electrocardiogram measurement, which shows the relaxation and contraction of cardiac muscles. This the LVIDS should be very narrow, but LVIDD should be wide open. So, but in SHR, it's a little bit different. They relax more and cannot contract much. But with hypoxia preconditioned stem cell, they are very effective. They almost look like the controls. And moreover, the cardiac size was also reduced with 6 hours of preconditioned stem cell transplantation. And here you can see that in hypertensive animals, the heart shows a thickened wall, meaning hypertrophied heart. But upon treatment with the preconditioned stem cell, the effect is very good. They reduce the uh, wall thickness just like the normal ones. So in adult animals, we see that 6 hours of hypoxia treated preconditioned stem cells give very good protection. And this is also true with mesenteric staining. With mesenteric staining, we can find the fibrosis, the collagen accumulation. So when there is more blue color shows the collagen accumulation. When there is more collagen accumulation, the cardiac contractile function will be lost. So that is obvious in the hypertensive animals, but upon treatment, they are reversed almost like the normal. But without preconditioned without stem cell administration, without preconditioning, we see that it's not that good. And with the preconditioning, it's good enough. And in this way, we also have in our hospital, we have a commercial product called MitoBurst. This MitoBurst controls the mitochondrial ROS. When this is a commercial product, we just have to culture the cells with MitoBurst. When you culture this, usually when you culture cells for more than 3 or 4 passages, the stem cells, they go for differentiation, and also the function will not be restored after 3 or 4 passages. But with MitoBurst, that can be like the pink liquid here is the MitoBurst, and with this MitoBurst, if you culture the stem cell regenerative potential, the stemness will be retained. And so we also find some preconditioning to improve this homing. Homing is based on chemokine signaling. Where damaged tissue, usually they produce some chemokines like the SDF1. SDF1 is a chemokine. A damaged tissue, they just send this as a signal, like SOS, they send SDF1. But stem cell contains some receptor they have receptors for this SDF1, like the CXCR7 and CXCR4. CXCR4 is a traditional receptor, and CXCR7 is a newly found receptor. And when we increase this receptors in stem cell, it is more likely that they can go towards the SD1 secreting damaged tissue. So we have found a in vitro model for aging using doxorubicin. Doxorubicin is a chemotherapy drug, but with low concentration of doxorubicin, they induce aging. So yes, like cardiac cells with the doxorubicin, they're treated with stem cells, and these stem cells, when preconditioned with AOF, AOF is a well-known Chinese drug for anti-aging effects. This drug, when we precondition the stem cells with this anti-aging Chinese medicine, we found that the CXCR7, or the receptor for homing, increased, and this helped the stem cell transplantation more efficient. So for this, to find this, we performed a multiple like a transfer assay. In a transfer assay, we have 2 chambers. In the lower chamber, we use we culture cardiac cells, and the upper chamber we use the stem cells. So this stem cell, if they move, there is a membrane that separates these 2 chambers, and when they can move to the opposite side of the membrane, it means the cell can migrate. So when we culture here, the cardiomyocytes here and the stem cells here, and if the stem cell can move towards the damaged cardiomyocytes here, it means their migratory ability is good. So now we found that when we treat this AOF, the AOF reduced the aging aging of the stem cells. So beta galactosidase is a very good marker for aging. When we treat doxorubicin, which is a aging inducer here, the beta galactosidase increased. But upon ADMSC, like upon stem cell treatment with AVOF preconditioning, the beta galactosidase reduced very well. And then also we tried the chemokine receptor, like we checked the chemokine receptor levels in the cell, and we see that ADMSCs, or the stem cell preconditioned with the AVOF, showed increase in the CXCR4 protein and also increase in CXCR7 proteins. This means the AVOF can has an anti-aging effect and also can enhance the stem cell mobility, migration towards the damaged tissue. So when we just transplanted these cells into rat models, we found that usually the collagen accumulation or the fibrosis, cardiac fibrosis that is seen in aging hearts was reduced when treated with AVOF preconditioned stem cells. Also the cardiac function, the EF, the ejection fraction and fraction shortening, they are the indicators for cardiac function. So this usually in the hypertension animals, it just goes down. The ejection fraction is very less than 70, but in upon treatment, the ejection fraction is regained. Also, the fraction shortening, usually it will be around 45, but in hypertensive animals, the fraction shortening goes is reduced, but upon treatment with the preconditioned stem cell, it increases. Also, we use other methods to improve the foldings, which the protein foldings are as I told, protein foldings, unfolded proteins, they cause proteotoxicity. But usually chaperones or proteins that usually bind to proteins and help to maintain their protein foldings or help to degrade the unfolded proteins. For example, an unfolded peptide, a synthesized peptide, it binds with HSP70, which is a chaperone, and it just tries to, it can maintain the proper folding of the protein and performance function. And in our lab, we see that CHIP, a co-chaperone of HSP70, to be more effective in maintaining protein proteostasis. For example, IGF-1R. IGF-1R is a very important receptor, insulin growth factor receptor in every cell. So IGF-1R is usually induced by IGF-1, ligand for the IGF-1R. This IGF-1 secreting stem cells seem to be more proliferative, and these stems. So now, in our study we found that CHIP, that's a co-chaperone, that can increase. It binds to the IGF-1R. CHIP can bind to the IGF-1R and maintain its foldings. We found this by a co-immunoprecipitation assay. With this co-immunoprecipitation assay, we pull down the CHIP from the cells. We pull down using antibodies. We pull down the CHIP proteins and then run in a gel and transfer to a membrane. Upon this, we found that and then check for IGF-1R levels. So IGF-1R level, if we can detect IGF-1R here, it means that this IGF-1R was bound to CHIP. So we found that upon CHIP overexpression, this IGF-1R level increased, meaning so CHIP increase can increase IGF-1R. So binding of CHIP to IGF-1R stabilizes IGF-1R. Otherwise, the IGF-1R will be degraded, and IGF-1R degradation may cause reduction in survival signaling. Survival signaling can be indicated by the levels of PAKT. PAKT level increases upon increased IGF-1R, meaning CHIP overexpression increases IGF-1R, which also increases AKT, or the survival mechanism. So then we also have strategies to enhance stemness in stem cells. For that, for example, the CHIP upon overexpression it also enhanced the transcription factors of stem cells, meaning they can maintain the stem cells. So they chip also degrades a protein that PTEN called PTEN that interrupt the survival signaling. The PI3K PAKT signaling is important in the is an important survival signaling that is blocked by PTEN, but chip overexpression can prevent that. Yeah. So we have formulated that chip overexpressing stem cell is a very good stem cell candidate for stem cell therapy among aging individuals. And then finally, we also checked exosomes. Usually, stem cells, they should be maintained, their expression levels is controlled, is tightly regulated by methylation and demethylation. Demethylation enzyme, DNA methyltransferase is a methylation enzyme that can methylate genes, and we found that microRNA 92A25P is a microRNA that inhibits DNMT3A. So increase of DNMT3A means the stemness of the stem cell is lost. They undergo differentiation. So when microRNA 92A25P is lost, when the level of microRNA 92A25P is lost, we found that the DNMT3A is increased. So now we are finding some strategies to inhibit this microRNA 92A5P so that we try to increase the levels of microRNA 92A5P, thereby to inhibit the DNMT3A level and thereby to restore the stemness of the cells. So these are the strategies we are now using in our laboratory and to find a better stem cell for regenerative potential. Yeah, thank you. So this is our group. We have a very strong group of 42 researchers here, and this is our boss, the president of this research center, Dr. Chiang Huang, and his wife. There are so many Indians in our group as well. Yeah. So thank you for your patient listening. And I've seen. Hello.
Hello, sir.
Yeah.
Hello. This is Samsavini, sir.
Yes,
so thank you for your nice presentation, sir.
Thank you.
So there are few queries in the chat box, sir.
Yes, I'll look into that.
Yeah. From Shivaranjani Ishwari, she has asked, is there any possibilities to cure COPD patient with stem cell treatment? Is there any possibilities to cure COPD patient with stem cell treatment? Chronic obstructive pulmonary disease.
So, there are hello. Yeah.
Sir, yes.
Yes. There are, of course, many strategies that are used in preclinical studies for COPD patients, and it's like there are I have seen that I have heard that about 82% under clinical trial they have patients that they have 82% of the people who have tried for stem cell transplantation has shown good results in that.
Hello, sir.
Okay. For COPD, there are clinical trials with stem cells for COPD, and about in one of the study which I've heard long ago, there are it's about 82% even respond well to the stem cell therapy for COPD.
Okay. Okay, sir.
Yeah,
So
yes, yes.
the other question from Shivaranjani Ishwari, what are all the major side effects the patients face then and there when they undergo stem cell treatment?
Sorry, can you come again?
Yeah. What are all the major side effects
Mhm.
the patients
Okay.
face?
Yeah. Okay, I got it. So
Yeah.
the major side effects, like initially, that's the most scaring thing about this was teratoma formation. Teratoma formation is cancer, like tumor growth. When like the patients, when they are transplanted with stem cell, they get like there may be a unfortunate tumor growth with that. But the another thing is the immune system dysregulation. So, for example, for in hypertensive patients, when we inject stem cells from the when we make some okay when for hypertensive patients, when we inject stem cells, it has been shown that the inflammation increases because these stem cells in that, for example, with the angiotensin 2 is very much associated with hypertension. In hypertensive patients, the angiotensin level increases. This angiotensin increases the differentiation of these stem cells, and they differentiate into hematopoietic cells, and they increase the inflammatory response in a system. So this is another problem in the patient. And again, also this ethical issues are also another problem. Ethical issues like people cannot accept something which is like I've heard the stories about even blood transfusion. People don't accept blood transfusion immediately. Only when they are pushed to a condition they accept that. In the same way, like stem cells also will have some issues on that. So people may not readily accept that. But so far the problem is the retention in the stem. Like the major problem is the retention of stem cells in a host system. When we inject stem cell, that should stay in this in the body for enough time so that it can be the damages should be repaired. So now there are so many, like to overcome this, there are now the approaches are like we just try to induce the uh, native stem cells, endogenous stem cells which a patient already has, so that to induce that stem cell itself and to because the because when you inject the stem cell and to avoid the host rejection, we just use immunosuppressants. So when we use immunosuppressants, the patient has to be in the hospitalized condition for a long time until the particular effect is achieved. So that's another issue. Yes.
Okay. Sir, question from Raga Niyantri. Sir, why is the stem cells injected through tail and why not in other routes of injection?
Tail because it is in animal model that I have shown, but usually for even for heart, pericardial injection can be done just in the heart, like can be done also in the brain. For brain, usually that is local. For human trials we majorly focus on localized injection, but for systemic injection, for systemic transplantation, because stem cell not like we have, as I mentioned, there are two ways of function for stem cell. One is stem cell just goes to the damaged area and then they differentiate there and replace the damaged tissues. For this localized uh, transplantation is injection is good. But in another case, the stem cells, they produce a lot of paracrine factors. These paracrine factors will be spread over the body to every tissues, and that's good for, like for example, for the condition like aging. This is essential. So when you want to regenerate a person, a systemic administration is essential. So we usually give by vein, like intravenous injection. So for when we saw like for intravenous injection, I think which will be like should be directly on the blood, not by. But there are, of course, there are some preclinical studies in smaller animals with IP injections. Also nasal for brain, they use nasal injections. like that, but intranasal injection. But generally, IV injection is good, is better. Like it can reach the bloodstream immediately and can reach the target tissues easily.
Okay, so next question. Meera, research scholar from Kongunad Arts and Science College. Why diabetes patients are excluded for stem cell therapy if they had further pathologies or changed endocrine status other than diabetes?
Why? I'll just read the question. Okay, why diabetes?
Yeah.
Yeah, I told, like, for example, that depends on this on the cells that we use. For example, the hematopoietic stem cell, they are not recommended for this, because they interrupt the endocrine system. For example, diabetic patients, if the patient, for example, in with the thyroid deficiency, will not will not be prescribed for this, because they interrupt the other endocrine system. That's an issue. That question, actually, the question itself has the understanding of this aspect here. So usually, but there are so many trials which undergo, like that is carried out for stem cell transplantation therapy in diabetes. For example, mm, like there are stem cell transplantation to improve the pancreatic function, to find the beta cell differentiation of pancreatic cells, the iPSC. So they have been tried, but only with specific kind of stem cells, they are not recommended, like the hematopoietic stem cells.
Okay, sir. So, a question from Chellamuthu. What type of stem cell population decline upon aging? MSC or HSC or both? And stem cell have infinity self-replication potency than other somatic cell, right? What is the dose of stem cell used to treat cardiac problem?
Okay, so usually MSCs can be derived from almost all tissue systems, and hematopoietic stem cell, as we told, it's maybe uh, usually from bone marrow. Usually, the stem cells, the bone marrow, usually they decline, and mesenchymal stem cell, it depends on the tissues where we get. For example, the bone marrow mesenchymal stem cells, they decline, but adipose derived mesenchymal stem cells, they it's preferred one, because it is easy to isolate. We have an inject, and we take the adipose from humans. For animals, we just do some small surgery and cut out the adipose tissue, and then we try to take the stem cell. But upon aging, that's not the mesenchymal stem cell population may be good, but they're not good enough for multipotency. They may differentiate sooner or later into adipose tissues or chondrocytes, or usually mesenchymal stem cells, they differentiate into chondrocytes, uh, also adipocytes, like that. So they differentiate into four types of cells, and we may not be able to achieve a particular desired effect when we take from an older patient. So usually it's not just only the population declines, but also the quality declines with the aging. And yes, stem cells have like, we cannot say infinity self-replication about stem cell. They are not infinite. It depends. Not all stem cell are infinite. For example, when we are born, we have immense the healthy stem cell pool. But when we grow, for example, heart, when you see, when we are born, for the first 3 or 4 days, the heart can proliferate. The cells in the heart can proliferate. But after 4 or 5 days, the proliferation of the heart stops, and all the cells became mature there. Like that, the stem cell in a particular tissue system, they will be there as with potency, but upon aging they just reduce. So there is no point of infinity there in case of MSc and HSCs. So for a dose, usually it depends. Usually in animal models we use 10 power 7 cells, 1 into 10 power cell, or 4 into 10 power cells in one dose, if it is a one dose, single dose. And yeah.
So, a question from Dr. Narendra Kannan, sir, of Kongnad Arts and Science College.
Yes.
How about the affordability for stem cell treatment?
Affordability, it's economically, it depends. So, like for example, the hematopoietic stem cell are done, or we are like it's performed in many hospitals. Even like in Bangalore, I don't know very much about India, but I know that in hospitals in Bangalore they do hematopoietic stem cells transplantation for immune related diseases, and also for cancer treatments. But of affordability, like I hear that like it's higher than what we use stem cell therapy only for those conditions which we don't have other cures. So like it depends on the if there is no other way, we have to go for it.
Okay. So, a question from Mohammed, sir. Can stem cell are useful in treating cancer tumors?
They are, of course, like the most of the, about more than 60-70% of the stem cell therapy is tried to cure cancers. And there are, of course, we have so many drawbacks in treating stem cell with chemo drugs. The same way we have some drawbacks, but now we have the CAR T cells, chimeric antigen T cells. This is very popular in, so we use some hematopoietic stem cell, our T cells. We just engineer their antigens so that they just target to a specific particular cancer type. So for targeted therapy, we use CAR T cells now. That's very popular and are being tried in clinical trials. But and the people have very high expectation. Previously, we were trying with PD1, PD-L1. This is suppressing hematopoietic stem cells were tried. These stem cells will go to the particular tumor environment and suppress the PD1, PD-L1. These genes are immunomodulatory genes. They just help the cancer cells to evade the immune system. So previously, stem cells stem cells which can remove these particular genes were tried, but finally somehow they failed. It was not working. But these CAR T cells are very promising.
Okay, sir. Question from TJ Jomi. Could you please comment on the success rate of stem cell research on bronchial dysfunctions, and is stem cell an alternative for knee replacement?
First of all, I should tell that I'm not very much expert on pulmonary diseases, but still, but about the bone, like also like knee replacements, among the success stories, stem cell therapy for osteoporosis is very successful. They have it's in very advanced stages, and there are so many people working on it, and they have used so many different systems to create the they have found some chaperones or stencils to find so that the stem cells can grow over that and can be used as a it can be used as a therapeutic option there. And there are it's in very advanced stage compared to other, for example, cardiac or neuro related therapy. Osteo researchers are very successful.
So she also asked whether the stem cell therapy is permanent.
That's very good question. Very good. Yeah, but we cannot say so. We cannot say that it is permanent in many cases. For example, aging, it cannot be permanent. And but for other things like osteo, like bone displacement, like bone related ones, and cardiac, as well as liver transplants. For that, I think they are good. Also, for example, the like immune dysregulation related ones, they are successful. They are permanent as well. Like bone marrow transplants, all these are very successful and almost permanent. But other things like, for example, the anti-aging treatment, I don't think that will be permanent. And yeah.
Okay. So a question from Karthik Kumar from Kongnadu College. How do the fission and fusion proteins differ in a post-translational modification of mitochondrial dynamics in stem cells?
Good. Actually, fission and fusion proteins also play a major role here. Yes, very good. Because stem cell, I told, like when stem cell at a quiescent stage, they do their metabolic rate is very low. So their mitochondrial level will be, the cells will be in a resting state. They don't need that many mitochondria. But but upon stimulation for differentiation and proliferation, and the cells has to proliferate more, they have to undergo mitochondrial like like they will have increased metabolism. So the mitochondrial level will be balanced. Mitochondrial fission and fusion will be elevated. But this also affects the stemness of the cell. When the fission and fusion are unbalanced, the ROS level, like the stress in the cells, will enhance, and they become like differentiate, and they terminate their proliferation.
Okay. Sir, shall we take few more questions, sir?
Sure.
Yeah. Okay. From Chellamuthu, sir, what is the percentage of biodistribution of stem cell in our organs on systemic administration?
Oh, okay. This is also a good, like, but usually the first place for accumulation is the lungs, and then to heart, if it is systemically administered. And then it depends on the, like, and then the blood vessels, like wherever there is damaged tissue, whichever cells that produce SDF1, high SDF1 level, the cells will accumulate there. I cannot guarantee on the timeline how long it will stay. We cannot say. Maybe just for a short time. But usually the SDF1 producing tissues, like most tissues, they produce SDF1. But still, heart, lungs, and the kidney, they attract more cells. And then the blood vessels. So these are the places where they get accumulated more, but not for long.
Okay. Question from Asha Kannan. Sir, is there any stem cell therapy for Alzheimer's disease?
Yes. Alzheimer's disease, we can, like, it's not so, like, many studies are in clinical study stages. There are many. Even in our lab, we are working on a complex like a complex pool of stem cells. We have generated complex pool of stem cells to treat Alzheimer's disease. We use some few kinds of cells from umbilical cord, few from adipose, and we try to form a strategy to improve the efficiency of stem cells, to increase the but still, I don't say it is in an advanced stage, but sooner or later there'll be it depends on the approval. For example, in our hospital, we are ready to provide this, but upon approval, we have got approvals for clinical studies, and we are doing it now.
Okay. So a question from TJ Jomi. What are the potential developments in stem cell research with regards to COVID-19 clinical trials and the futuristic approach towards such pandemic undermines?
Yeah. But like stem cell can be used to boost the immunological response. Hematopoietic stem cells can be transplanted to boost the immunological response. That's one point. But otherwise, the major targets of COVID-19 is lung and heart. So we have already studied so many strategies to protect the cardiac tissues and lungs. So those kind of strategies can be tried, but but still this is in a very preliminary stage, because I don't think there are clinical trials that are involved to treat viral infections. I know that the after effects of viral infections, uh, we have strategies, or strategies are being tried for after effects related with viral infections. But no one would have hoped that a viral infection would need a stem cell therapy so far, but can be tried from now on.
Okay. A question from Meera Darshini. Stem cells has the features of self-renewal and differentiation. Paradoxically, it has the potential to form cancer cells. What should be the precaution taken during therapy in considering the risk and their benefits as priority?
So usually the cells when transplanted, those cells which can live shorter life, not for long, can they have very less risk uh, for tumor formation. Those cells which have, so one thing can be done. We should not expect like a stem cell that should go there and stick to the damaged tissue, or for the purpose, and just stay there to find the to give a expression. But we can have the paracrine effects. For example, we have found so many exosomes of stem cells. These exosomes, they can they actually carry out the paracrine functions of stem cell. In many cases we have found. So we isolate the exosomes and inject the exosomes instead of the stem cell itself. And these exosomes can go and transfer whatever paracrine factors that are secreted by the stem cell into the target cells. This is another very good approach now, and it's been very well studied now. And yeah, this is one way to overcome that. Otherwise, we have very less differentiating stem cell, very less retentionable stem cell can may have lesser risk for cancer. So there should be a balance between that. It should not be that when you inject the stem cell, that should be infinite. There should be only a time frame for that.
Okay, sir. Question from Joshua Stephen. Um he has written a big paragraph. Since there is a fair risk of the injected stem cells transforming into cancer stem cells, and since the genetic makeup with respect to the immune system components of the host plays a role in this, sequencing both the host cells as well as the stem cells would be a very useful precaution. But the costs are massive. Can we design an ex vivo mixed culture method of both these cell lines, checking the compatibility, etc., before injecting it?
Yeah, it's a good way, but I don't think. Yeah, it's the genetic makeup, of course, plays a major role, but but I don't know whether the stem cell they form they need. Yeah, like okay, I can understand that question. The stem cells, like at this stage, we don't think we just take a stem cell and just inject. Not like that. We make a stem cell pool. We make a stem cell pool that is compatible, and from like that, when we do so many things to find out whether these stem cells are really ready to be transplanted. Not all cells will be transplanted into stem cell. For example, a colleague in our hospital, he has found that cells that are overexpressing beta catenin, they tend to be more like they tend to be to cause more cancer phenomena. So what he does is he removes, like when we collect the stem cell, he removes the beta catenin overexpressing cells, and he just segregates only those cells with lesser beta catenin levels and he injects. So this is one way to do it. And to actually now The focus of many research group is to find a universal stem cell pool. You, so that this universal stem cell pool can be injected to anyone, just like getting a crocin and having. So you just work for this treatment. We need this stem cell. So far, it is not that like we have the research has not gone to that level, but that is the motto. That is the goal. To find a universal stem cell pool to inject, so that so for this, of course, those studies should be done in a vast scale, whether this particular stem cell pool is compatible to all like every individual, not as a personalized medicine, but as a community medicine. Can it be used? It has to be checked, but it's a good thought.
So, a question from Karthik Kumar.
Mhm.
What is the current and future role of stem cell research in the conservation aspects to the wildlife community?
It's stem cell can be thought to like can be could be considered for, because even while when we do the preclinical test, we do it in animals. So of course there can be uh, chances for like extending the stem cell research to other aspect as well. Uh, but should be tried on like for like the people who are interested in that field should try that. But based on the intensity of work involved, for example, the infrastructure that is required and the financial need that is required to bring out these studies are very huge. For example, I will say our group has a standard funding of 2 million every year. So we require, and in addition to the seasonal fundings, we have a fixed funding of 2 million. Sorry, 20 million, not 2 million, 20 million. So this is required. So, and not just us, there are so many numerous groups that are working on that. So if some group that are very, like interested in wildlife, like expanding the stem cell therapy for wildlife community, it is good to do that.
Okay, sir. Thank you, sir.
Thank you.
I think we have clarified all the queries.
Thank you. It's a pleasure to
So,
join.
sir, thank you so much for
Thank
the
you.
nice presentation and clarifying all the queries.
Thank you.
And sir, now Dr. V. Bhuneshwari ma'am, I welcome Dr. Bhuneshwari ma'am to share few words. Dr. Bhuneshwari
Okay.
ma'am.
Yes, ma'am. It's a really wonderful and information session on stem cell therapy. Dr. Shibu, you had given a real research point of view towards stem cell therapy. So I'm very much proud that you have proved that you are not only a leading stem cell scientist, but you had explained well to the public also. Thank you very much. I hope you will accept the collaboration with our college to develop a stem cell lab in our department. Thank you very much.
Thank you. Thank you.
Thank you, ma'am. Sir, you highlighted about stem cell types, stem cell transplantation, age-related changes in the central nervous system, stem cell therapy for Alzheimer's disease, major clinical hurdles for routine clinical implementation, stem cell homing, strategies to improve the function of aging stem cells, hypoxia preconditioning to improve the stem cell potential. Thank you so much, sir. And now, as a token of love and gratitude, the PG and Research Department of Biotechnology, Kongunadu Arts and Science College, feels proud to honor our resource person, Dr. M.A. Shibu, sir, with an E-certificate. So kindly accept it, sir.
Thank you.
Yeah. Okay. So the same will be emailed to you, sir.
Oh, okay.
Thank
Thank you.
you.
Thank you. Thank
Thank
you.
you. Thank you, sir. Okay, so with this happy moment, now may I request Dr. B. Vishnupriya ma'am, Assistant Professor of Biotechnology, Kongunadu Arts and Science College, to express her words of gratitude. Ma'am, please.
Yes, ma'am. Ma'am, is it audible?
Yes, ma'am. Yes, ma'am. Proceed.
Yes, ma'am. A very good evening to one and all present here. It's my great privilege to propose a vote of thanks on this third session of International Alumni Webinar Session organized by Kongunadu Arts and Science College. This is a very important occasion for us. It is a great pleasure to present our words of thanks. Thank you very much. 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 Than
Vishnupriya ma'am, you're not audible, ma'am.
By Department of Biotechnology, Kungunadu Arts and Science College. I take this opportunity, Dr. C.A. Vasaki, ma'am. Is it clear now? Hello?
Yes, ma'am. Yes.
Hello?
Yes. Yes.
Hello?
Yes, ma'am. Yes, ma'am.
Not audible?
Audible, ma'am.
I take
Proceed.
Okay, ma'am. I take this opportunity to thank our honorable. I take this opportunity to thank our honorable president, Dr. M. Archami sir, and our beloved secretary and director, Dr. C. A. Vasaki ma'am, for your immense support and encouragement rendered in the growth of the department. Thank you, ma'am. I am grateful to thank our honorable principal, Dr. M. Lakshmanaswamy sir, our distinguished chief executive officer, Dr. V. Chinnaswamy sir, our respected Dean R&D, Dr. S. Palsani sir, and our beloved HOD Biotechnology, PG, Dr. S. R. Madan Shankar sir, and Dr. V. Bhuneshwari ma'am, Biotechnology, UG, for their moral support and guidance to organizing this webinar series in this pandemic situation. Once again, thank you all. On behalf of Kungunadu College of Arts and Science and the Department of Biotechnology, I extend my sincere thanks to the chief guest of today's session, Dr. M.A. Shibu sir, for making excellent presentation on stem cell therapy for aging associated disorders. Thank you, sir. Also, I would like to extend a special thanks to organizing secretary, Dr. R. Amsaveni ma'am and Dr. M. Gogul Ramnath sir, assistant professors in biotechnology, session coordinators, and also thank student volunteers. Thank you all for making this event successful with your contribution. I also wish to express my big thanks to all the participants for their active participation in this event. Thank you all.
Thank you, ma'am.
Thank you, ma'am.
A gentle reminder to all the participants. Continuation of this webinar series is on tomorrow, that is 7th June. And our resource person is Dr. M. Giriram Kumar, postdoctoral fellow, Center for Integrative Genomics, University of Lausanne, Switzerland. And the topic of discussion is the Drosophila 7SK SN-RNP complex is required for synaptic growth and function of motor neurons. So I request all the participants to join us by 10:45 a.m. tomorrow with the same Google Meet link. Thanks for each and every soul behind this program. With this note, I conclude today's session. Stay happy, stay healthy. Thank you all. Thank you, Dr. Shibu sir.
Thank you, sir.